All right, so today we're diving into something pretty wild.
Yeah,
We're talking about unboiling an egg white.
you heard that right.
It sounds like, pure science fiction.
It really does.
But we've actually got some really fascinating sources here, starting with a peer-reviewed paper from Biochem.
Excellent source.
There's also a great article that explains the research from UC Irvine.
Okay.
And get this, we even have a YouTube video that actually shows this seemingly impossible process, like actually happening.
Wow, that's pretty amazing.
It is, right.
Yeah, and so what I think is really cool about this is that it's not just like a cool trick, you know what I mean?
Right,
Well,
sure.
like beyond the visual of it, the thing that's so remarkable is the implications for how we actually produce proteins.
Yeah, exactly.
And so just think about it for a second. Like the huge demand for protein is everywhere.
Oh, absolutely.
So we're talking about things like life-saving cancer therapies,
Oh, yeah.
like the food we eat, and even just like the fundamental research that underpins it all requires proteins.
Yeah, that's such a good point. The world of biotechnology is just massive. And like you said, like a huge part of it all comes down to making large quantities of very specific proteins.
Exactly, yeah.
And that's where things get really tricky.
It does get tricky, yeah.
Because, as our sources kind of highlight, when you try to use these fast-growing and cost-effective organisms like E. coli, which is a bacteria, everyone's heard of, or yeast,
Right, right.
you often run into a major, major problem.
Yeah,
These proteins, they don't always fold properly, and
Yeah.
they clump together into these like insoluble masses that are called inclusion bodies.
and that's a real pain.
Yeah, and this is where traditional protein production, hits a wall.
It really does.
Because as the paper kind of goes into detail about, it's really slow and expensive to get correctly folded functional proteins out of these inclusion bodies.
It's a nightmare.
Yeah, we're talking about days and days of dialysis using just enormous amounts of liquid. I mean, literally liters and liters,
Oh.
just to get these tiny, tiny amounts of usable protein.
Yeah, you're right.
It's really inefficient, and the UC Irvine article actually points this out too. It mentions that these like older methods can take the equivalent of 4 days at the molecular level.
Oh, wow.
So, it's a huge bottleneck.
I see. Yeah, that's a huge problem.
And the alternative, like for really, really valuable or like super complex proteins, sometimes you have to use these like much more finicky and expensive cell lines, mammalian or insect cell lines,
All right.
which is not always feasible, especially when you're trying to scale up, for large scale production.
So this is really a huge problem for the industry.
It is.
Yeah.
So scientists have been trying to find more efficient ways to solve this whole protein folding challenge for a long time.
Right.
And the paper does mention one existing approach,
Uh-huh.
which uses hydrostatic pressure. But what we're really diving into today is something completely different.
Okay, I'm intrigued.
Yeah, it's a completely novel method that uses something that's actually surprisingly gentle,
Okay.
but super effective, and that is shear stress.
Shear stress. Okay, let's unpack that a little.
Okay, yeah, so let's talk about shear stress. The big star of the show here is a device called, are you ready for it? It's called the Vortex Fluid Device,
Uh-huh.
or VFD.
All right.
Yeah, and I know, it sounds super high-tech.
Very fancy.
Yeah.
Yeah.
But the idea behind it, as our sources kind of explain, it's actually really elegant.
Okay.
It is.
Right.
So at its core,
Uh-huh.
it's basically a glass cylinder that spins incredibly fast.
Okay.
You're talking about like 5,000 revolutions per minute.
Wow. So fast.
Yeah, crazy fast.
Okay.
And here's the catch. It's also tilted at a 45-degree angle.
I see.
So because of that tilting and the spinning, it creates this super thin film of liquid.
Okay.
We're talking like just a few micrometers thick along the inside of the cylinder. And because the cylinder wall is moving so fast and the liquid is right there up against it,
Uh-huh.
it creates this difference in speed within that super tiny film of fluid.
Yeah, I get it.
And that's the key, right?
Right.
That internal like kind of smearing force
Okay.
where different layers of the fluid are moving at slightly different speeds, that's basically what shear stress is.
That's molecular level, right?
Exactly, yeah.
Yeah.
And the paper even gets specific.
Okay.
They calculated the range of shear stress at 5,000 RPM,
Okay.
and it's somewhere between 0.52 and 0.56 pascals.
Pascals. Okay.
Now, I'm not a physicist, so, I don't know about you, but
Me neither.
I'm wondering,
Yeah.
what does that actually do to the protein?
Right. Like, what does that mean?
Exactly.
Yeah, that's the big question, right?
Yeah.
So what's interesting, and the paper points this out, is that these levels of shear stress are actually similar to the levels that have been shown to cause proteins to unfold.
Oh.
Yeah, so what seems to be happening is this kind of like a dynamic process of this constant, gentle unfolding and refolding.
Oh, interesting.
Yeah, so it's almost like imagine, the misfolded proteins are just being kind of nudged
Okay.
until they can like find and then settle into the correct shape.
Okay, so they're not being forced.
Yeah,
They're just being like kind of
it's like a gentle persuasion.
encouraged.
Yeah, yeah, exactly. Like think of it like this. It's like, when you have a crease in a piece of paper
Okay.
and you're trying to get it out.
Uh-huh.
You kind of work it gently, right?
Yeah.
You don't just like jam it flat.
Right,
You
right.
kind of like work it until it flattens out.
That's a great analogy. Yeah, I get that.
Yeah.
It makes a lot more sense.
Yeah, yeah.
And you know what's so cool? This same VFD technology has actually been used for a lot of different things.
Really?
Yeah, it's really versatile. So they've used it for things like breaking down tiny little molecular containers.
Uh-huh.
They've also used it to create smaller particles for like brand new materials.
Wow.
And even get this, exfoliating graphite.
No way. That's really cool.
It is, right. So it really highlights how versatile this whole controlled mechanical force can be.
Exactly. And so I think the researchers, they saw this, right?
Yeah.
They saw the potential of this and thought, okay, what if we apply this gentle but controlled energy to this protein misfolding problem?
It's really clever. Yeah.
And I think the thing that really captured the public's attention, when this paper came out,
Yeah.
was their initial experiment
Uh-huh.
that used something that, that everyone can relate to.
Right. A boiled egg.
Yeah, the unboiling demonstration.
Exactly. That's what everyone's talking about.
That's very cool.
So they took, a hard-boiled egg white,
Uh-huh.
which is basically just this solid white mass of like totally tangled proteins. Right.
Yeah.
And the first thing they did, which, you can actually see this in the YouTube video,
Oh, cool.
is dissolve it using urea.
Urea, okay.
And so urea is a chemical that basically it unravels the protein structure
Yeah.
and it turns that solid egg white back into a liquid.
Right.
But the proteins are still misfolded at this point.
Gotcha.
So then they take this urea solution,
Mm-hmm.
they dilute it down,
Okay.
and then they pass it through the VFD.
Right. So this is where the magic happens.
Exactly. And this is where it gets really interesting. The paper says that after just 2.5 minutes,
2.5.
I know, crazy, right?
Oh.
2.5 minutes of processing in the VFD at 5,000 RPM,
Okay.
they were able to recover the activity of lysozyme.
Okay, and lysozyme is?
So lysozyme is a key protein that's found in egg whites.
Got it.
And that is crazy fast compared to how long it would take using traditional dialysis.
So we're talking, days versus 2.5 minutes.
Exactly.
Yeah.
Yeah. It's really remarkable. 2.5 minutes is barely enough time to make a cup of tea.
Right. It's amazing.
I know, it's mind-blowing. But the paper does say that, you can actually apply too much shear stress.
Oh, interesting.
And that can cause the protein to just unfold again.
Oh, so you have to get it just right.
Exactly, yeah. Finding that, Goldilocks zone, the optimal conditions, is really important.
I see.
And for the egg white lysozyme, they found that 5 minutes at 5,000 RPM in a slightly less concentrated urea solution was like the perfect sweet spot.
I see. So they had to tweak it a little bit?
Yeah, exactly. Just a little bit.
Okay.
And they didn't stop there.
Okay.
They didn't just do the egg whites. They wanted to see if this would work with other proteins as well.
Okay.
So they also worked with lysozyme that had been made in the lab using bacteria. So it's recombinantly produced.
Got it.
And this lysozyme, they actually deliberately misfolded it into those inclusion bodies that we were talking about. Oh, okay. So they basically forced it to misfold.
Exactly. Yeah.
And they followed a pretty similar process. They denatured the protein with urea, diluted it quickly, and then they ran it through the VFD.
Okay.
And the results, they were really impressive.
Let's hear it.
So the paper shows that after the VFD treatment, this lab-made lysozyme actually regained its proper, secondary structure.
Okay.
And they were able to confirm that using this technique called circular dichroism.
Okay, so what is that?
It's basically a way to, see the 3D shape of the protein.
Oh, cool.
Yeah, so they could actually tell that it had folded back into the correct shape.
Okay.
And what's even more important is that they measured the enzyme's activity. And they found that it had recovered over 82%.
82%.
Yeah, of its functional activity.
Wow. Okay, that's really good.
Compared to the like naturally occurring protein.
Yeah, that's amazing.
And that's like a huge deal, right?
It is, yeah.
Yeah. And to take things even further, they actually developed a continuous flow version of the VFD.
Okay, so explain that. Like, what does that mean?
So imagine a system where you're constantly feeding in your solution of misfolded protein.
Okay.
And on the other end, you're collecting the correctly folded protein.
Oh, wow.
Yeah. So it's like an ongoing process.
Like an assembly line.
Exactly, yeah.
Very cool.
And the paper says that they get really similar, like high recovery rates with this continuous flow method.
Oh, that's really promising.
Yeah, it really shows the potential for scaling this up to meet the needs of, like industrial production.
Yeah, this is big.
It is.
So we've seen it work with lysosome now in a couple of different contexts.
Yeah.
But what about other proteins?
Yeah, good question.
Are there proteins that are known to be particularly difficult to refold?
Right, that's a great question. And the researchers actually, they took on one that's like notorious. It's called caveolin delta TM, and this protein is known for needing a really, really long time to refold.
Like, how long are we talking?
We're talking days using traditional dialysis.
Oh, wow.
Yeah, so this is where the speed and efficiency of the VFD, it really shines.
Okay.
So they did a short 1 hour pre-dialysis step just to lower the urea concentration.
Right.
And then they treated this caveolin protein with the VFD for just 30 minutes.
30 minutes.
30 minutes, yeah.
Wow, that's incredible.
I know.
Okay.
And again, they used that CD spectroscopy technique, and they saw that the protein's characteristic helical structure was restored.
Oh, very cool.
And what's interesting is that the solution also became a lot clearer.
Oh, okay.
Which suggests that those big clumps of misfolded protein were being broken down and refolded into soluble functional forms.
Oh, that's neat. Yeah.
Yeah, so it's you can actually see it working.
That's very cool.
Yeah, it's not just numbers on a graph. It's you can actually visually see the difference.
Right.
And what's really crucial here is that they showed that this refolded caveolin protein actually worked.
Okay. So how did they show that?
So they did these laboratory tests, and they showed that it could bind to its partner protein,
Okay.
which is called HIV GP41.
Okay.
And the strength of that binding was way higher in the VFD-treated samples compared to the ones that were either left untreated or refolded using traditional, long dialysis.
Right.
So it's not just making the protein look right. It's actually helping it to work correctly again.
That's amazing.
It is. It's really cool. But they did hit a little bit of a snag when they tried to refold even larger proteins.
Okay.
Like this one called, it's a mouthful, but it's CANMP-dependent protein kinase.
Oh, okay. We'll just call it PKA.
PKA, yeah, much easier.
All right. So what was the problem with that one?
So PKA is a lot bigger.
Okay.
And it's more complex than lysozyme and caveolin.
Yep.
And so the standard VFD protocol, it wasn't as effective for this bigger molecule.
I see.
Yeah.
So what did they do?
So they didn't give up, and they actually adapted the VFD method in a really clever way.
Okay. I like clever solutions.
Yeah, me too. So they were trying to mimic how proteins naturally fold inside cells.
Oh.
So they use this thing called a his tag, which is imagine a molecular handle.
Okay, like a little handle. Yeah.
Yeah, exactly, that they can attach to the protein.
Okay.
And then they use this special resin that this handle sticks to.
I see.
So it's like they were grabbing onto one end of the unfolded protein, and then they subjected the rest of it to the shear stress in the VFD.
Okay, so they're kind of directing the force to a specific part of the protein.
Exactly, yeah. It's like they're guiding the folding process.
Very cool.
Yeah, it's really neat. And after treating the PKA while it was attached to the resin in the VFD, they released the protein, and they found that it had recovered a significant amount of its normal activity.
Like how much are we talking?
69%.
69%. Okay, so not quite as high as the lysozyme,
right?
But still pretty good.
Yeah, it's still really good, especially for a protein that's so much bigger and more complex.
Absolutely.
And they did all these control experiments to make sure that it was really this targeted approach that was working.
Oh, good.
So when they used resin that couldn't bind the his tag, or they blocked the binding in other ways, they saw very little enzyme activity
So
recovery.
that really kind of proves the point, right?
It does. It really suggests that applying that sheer stress to that specific part of the protein, the N-terminus,
Yeah.
that was key.
That's really cool.
It was. It was.
So if we, zoom out a little bit and look at the big picture, right?
Yeah.
What are the most significant takeaways, like, for our listener?
Yeah, that's a great question. So the paper, it highlights some really, really compelling advantages of this whole VFD method.
Okay.
I think, first and foremost, it's the amount of liquid that you need.
Right. Yeah.
It's just drastically reduced.
So how much less, like, are we talking about, like, orders of magnitude less?
They're using about 1% of the volume.
1%.
1% compared to traditional dialysis.
That's incredible,
It's
yes.
huge. Yeah.
That's a game changer.
Yeah, and then the speed, too.
Oh, yeah, we talked about the speed. It's unbelievable.
It's mind-blowing. I mean, we're talking about minutes instead of days.
Yeah, yeah.
The paper, it says that it's over 100 times faster than overnight dialysis.
Wow.
And for that really challenging caveolin protein, over 1,000 times faster.
It's hard to even fathom that.
I know.
Yeah.
So just think about what that means for, like, time savings, cost savings, for research, for industry.
It's huge.
Yeah.
It's massive.
So it's faster, it's uses way less liquid, it's what else? Yeah, so on top of all of that, it's actually a relatively low energy process.
Okay.
And it's inexpensive.
Oh, even better.
And it's something that can be implemented in most standard biochemistry labs.
So you don't need any, like, fancy specialized equipment?
No, you don't need that high pressure equipment. It's basically just a spinning glass tube.
That's amazing.
Yeah, it's really simple in principle, but it's so effective.
So this has the potential to, like, really, like, revolutionize how we make protein.
It does. It really does.
Wow.
The researchers, they're suggesting that this could be a whole new way to produce proteins.
Okay, how so?
So, like, you could deliberately overexpress proteins in simple cell lines and get the maximum yield,
Okay.
even if it means that you end up with those inclusion bodies,
Right.
because now we have the VFD that can efficiently refold those aggregates.
Oh, I see.
So it's like flipping the script
Totally, yeah.
on the traditional approach.
Yeah, normally you're trying to avoid the misfolding in the first place, right?
Exactly. Yeah.
And you end up sacrificing yield.
Exactly.
But now you're saying, like, we can just, like, go for it.
Go for it. Yeah. Get those high yields and then we'll fix it later.
That's a big shift in thinking.
It is. It really is.
And so, these lab scale results are incredible.
Yeah.
But what about, scaling this up?
That's a great question.
Can we use this technology to handle the huge volumes that you would need for industrial applications?
Right. Right.
Like, how do we go from the lab to the factory?
Yeah. And the paper actually does talk about that.
Okay, good.
They talk about the possibility of using continuous flow VFD systems
Okay.
or even just running multiple VFD units in parallel
Right.
to process much larger quantities of protein.
So it sounds like it's possible.
It does, yeah. And what's really cool is that they can actually change the design of the VFD sample tube
Okay.
to fine-tune the shear forces even more.
Oh, wow. So there's a lot of room for optimization and playing around with the parameters.
Exactly, yeah. So it's a really exciting area for, like, further research and development.
Absolutely.
And the researchers themselves, they're, like, super excited about the possibilities.
Yeah.
They really see this as a new set of tools for working with proteins.
I mean, it's amazing to think that something so simple, like mechanically, can have such a big impact on these molecules.
It is.
It kind of challenges our thinking, right?
Yeah.
About how we fold proteins.
It does.
We usually rely on, like, chemical methods or temperature.
Right.
But this is something totally different.
Yeah, it's really highlighting the importance of these physical forces that, like, maybe we've overlooked in biological systems.
Right, exactly.
So to sum it all up for our listeners,
Yeah.
the big takeaway here is that we now have a really, really simple yet incredibly powerful way to refold proteins.
And these were proteins that were previously, like, really difficult to work with.
Exactly.
Like, impossible almost.
Yeah, and the egg white demonstration, while it's, super visually cool,
It's a great visual, yeah.
it really does, like, show the potential of this technology.
Yeah.
It's like, it's not just a cool trick. It could actually change how we produce these essential proteins for medicine, for industry, for basic research.
Yeah, and if we, think about this in terms of, your interest in learning things quickly and efficiently.
Right.
The VFD itself is It's kind of like a shortcut
Oh,
in
interesting.
this really complex biological process.
Right.
It bypasses all those, time-consuming and resource-intensive steps of traditional protein refolding.
Right.
And it gives us a much faster and more streamlined approach to getting these proteins that we need.
I like that. Yeah, it's like a shortcut to knowledge.
Exactly, yeah.
So here's a final thought for our listeners to chew on. So we've seen that this technology can reverse something that seemed irreversible, the denaturation of proteins.
Right.
What other processes
Ooh, big question.
at the molecular level
Yeah.
might we be able to manipulate using these really carefully controlled physical forces?
Wow, that's a really interesting question.
Yeah, like what other possibilities are out there?
Yeah, and what other things that we thought were fixed, might not be so fixed after all.
Exactly. Yeah, it really makes you think.
It does, yeah.
So thanks for taking this deep dive with us.
Absolutely, it was a pleasure.
It was fun.
Yeah, it was a good one.
All right, until next time.
See ya.
Bye.